The growing demands for food security in a rapidly changing climate necessitate innovative solutions to enhance crop resilience. Polyploidy induction has emerged as a revolutionary strategy in plant breeding, offering a means to improve stress tolerance, yield potential, and adaptability in staple crops. This process involves artificially increasing the chromosome number of plants, mimicking a naturally occurring phenomenon that has played a significant role in plant evolution. By understanding the mechanisms of polyploidy, researchers and agronomists are exploring its applications in countering abiotic and biotic stresses that threaten global food production. The ability to create polyploid crops has opened new possibilities in agriculture, particularly in enhancing drought tolerance, salinity resistance, and pathogen defense in crops like wheat, rice, maize, and potatoes. This documentary delves into the science behind polyploidy induction, its historical context, and its potential to revolutionize agriculture by mitigating the effects of environmental stressors. Polyploidy refers to the condition in which an organism possesses more than two complete sets of chromosomes. In plants, this phenomenon is categorized into autopolyploidy, where multiple chromosome sets originate from a single species, and allopolyploidy, where chromosome sets come from hybridization between two different species. The process of artificially inducing polyploidy involves the use of chemicals such as colchicine, oryzolin, and trifluralin, which disrupt spindle fiber formation during mitosis, preventing chromosome segregation and leading to chromosome doubling. This genetic modification confers several physiological and morphological advantages to crops. Polyploid plants tend to exhibit larger cell sizes, increased biomass, enhanced photosynthetic efficiency, and greater metabolic activity. These traits are particularly valuable in improving resistance to abiotic stresses such as drought, heat, and salinity. For instance, research on tetraploid rice, Oryza sativa L, has demonstrated improved drought resilience due to enhanced root system architecture and increased water use efficiency. Similarly, polyploid maize varieties have shown greater tolerance to temperature fluctuations, making them more adaptable to climate variability. Many of today's staple crops, including wheat, cotton, and potato, are naturally polyploid. This characteristic has contributed significantly to their evolutionary success. The hexaploid genome of modern bred wheat, for example, arose through a series of hybridization and chromosome doubling events. This allows it to exhibit superior agronomic traits compared to its diploid ancestors. Similarly, cultivated cotton, an allopolyploid species, has gained fiber quality and yield advantages due to its polyploid genome structure. The artificial induction of polyploidy seeks to replicate and enhance these natural benefits by selectively increasing chromosome numbers in diploid crops that do not inherently exhibit polyploidy. Research conducted on diploid and tetraploid barley, also known as Hordeum vulgare, has revealed that induced polyploids demonstrate improved drought tolerance. This is due to changes in stomatal density and reduced transpiration rates. Such findings underscore the potential of polyploidy as a genetic tool for adapting crops to harsh environmental conditions. Drought and heat stress are among the most pressing challenges in global agriculture, affecting staple crop yields and food security. Induced polyploidy has shown promise in mitigating these stress factors by altering physiological and biochemical pathways. Studies on tetraploid maize, also known as ZA maize L, have demonstrated that polyploid plants exhibit deeper root systems, increased proline accumulation, and higher antioxidant enzyme activity. All of these factors contribute to improved drought tolerance. Similarly, research on tetraploid soybean, or glycine max, has indicated a significant increase in heat shock protein expression. This enhancement boosts the plant's ability to withstand elevated temperatures. Polyploid wheat lines have also shown improved heat tolerance through modifications in membrane stability, increased chlorophyll retention, and higher seed germination rates under heat stress conditions. These physiological adaptations not only improve plant survival but also sustain yield stability in drought-prone and high-temperature environments. Soil salinity is a major constraint to agricultural productivity, affecting nearly 20% of the world's irrigated lands. Polyploidy induction has been explored as a strategy to enhance salt tolerance in crops by modifying ionic balance and osmotic regulation mechanisms. Research on tetraploid rice has revealed improved salt tolerance due to increased sodium ion exclusion and enhanced potassium retention, reducing the toxic effects of saline conditions. A comparative study on diploid and tetraploid tomato, also known as Solanum lycopersicum, found that polyploid plants exhibited greater salt stress resilience by accumulating higher levels of osmoprotectants such as glycine, betaine, and trehalose. These compounds play a crucial role in maintaining cellular homeostasis under salt stress. Additionally, tetraploid barley has been shown to possess a more robust antioxidant defense system, which helps mitigate oxidative damage induced by salinity. Pathogen resistance is another key advantage conferred by polyploidy induction. Several studies have demonstrated that polyploid plants exhibit increased expression of defense-related genes, leading to enhanced resistance against fungal, bacterial, and viral infections. In wheat, polyploid varieties have shown higher resistance to rust diseases caused by Puccinia spp. A study on tetraploid banana, Musa spring. Similarly, polyploid cotton has exhibited increased resistance to Verticillium dahlia, a soil-borne pathogen that significantly reduces fiber yield and quality. These findings suggest that polyploid breeding could serve as a sustainable approach to reducing pesticide dependency while enhancing crop resilience against emerging pathogens. Apart from stress tolerance, polyploidy induction has been linked to improvements in crop yield and nutritional content. Polyploid fruits and vegetables often exhibit increased size, enhanced flavor, and higher concentrations of beneficial compounds. In strawberries, also known as Fragaria x ananassa, octoploid genotypes have been associated with higher sugar and vitamin C content compared to their diploid relatives. A study on tetraploid quinoa, or Chenopodium quinoa, found that polyploid plants exhibited greater grain size, higher protein content, and improved amino acid profiles. This makes them nutritionally superior. Similarly, polyploid wheat has been shown to contain higher levels of micronutrients such as zinc and iron. This addresses concerns related to micronutrient deficiencies in staple food crops. Despite its numerous benefits, polyploidy induction presents several challenges that must be addressed for widespread adoption in crop improvement programs. One major limitation is the difficulty in stabilizing polyploid genomes, as chromosome doubling can sometimes lead to irregular meiosis, reduced fertility, and genetic instability. Additionally, polyploid plants often require optimized agronomic management practices to realize their full potential in field conditions. Recent advances in genomic and biotechnological tools have provided new opportunities to refine polyploid breeding strategies. CRISPR-based genome editing, for instance, is being explored as a means to manipulate specific genes in polyploid crops, allowing for targeted improvements in stress tolerance and yield traits. Furthermore, transcriptomic and proteomic analyses are shedding light on the molecular mechanisms underlying polyploid adaptation, enabling researchers to design more resilient crop varieties. The integration of polyploidy induction with other breeding techniques, such as hybridization and marker-assisted selection, holds promise for accelerating crop improvement efforts. With the increasing need for climate-resilient agriculture, polyploid breeding is expected to play a crucial role in ensuring food security in the coming decades. Polyploidy induction represents a powerful approach for enhancing stress tolerance, yield stability, and nutritional quality in staple crops. By mimicking natural evolutionary processes, artificial polyploidy has demonstrated its potential to improve drought resistance, salinity tolerance, and pathogen defense in a wide range of agricultural species. While challenges such as genome instability and fertility issues persist, ongoing advancements in genetic engineering and molecular biology are paving the way for more efficient polyploid breeding techniques. As global agricultural systems face mounting pressures from climate change and population growth, polyploidy offers a viable solution to developing resilient crop varieties that can thrive in adverse conditions. The continued exploration of this phenomenon will be essential in shaping the future of sustainable food production.